Hi, we hope so that you will be fine. We hope that you will be familiar with the AlphaFold 2.0. This AlphaFold version has revolutionized the structural bioinformatics and greatly expedited the biological research, especially the pharmaceutical one. In previous video tutorial, we have explained it that how you can use the AlphaFold 2.0. Now in this video tutorial, we will explain you that how you can use the AlphaFold 3. The AlphaFold 3 is released in the start of May 2024 and got the eyes of researchers. So without any further delay, let's begin this video tutorial and let's see that how you can use the AlphaFold 3. To access the AlphaFold 3, you have to use a URL alphafoldserver.com. When you will use that URL, then you will be landed on this page. On this page, you can get an information about the AlphaFold 3 different type of the features. To access the AlphaFold 3 server, you need to click here on this server. When you will click here on this server, then you will be on this page. And here on this page, you can use the AlphaFold 3 to make a predictions of your three-dimensional structure of protein. The first very important thing that you need to know it here that how many jobs you can run on this server in today's date. As you can see it here that right now we have a 20 jobs quota. But this 20 jobs quota is dependent upon the tokens. As you can see it here that the jobs can be up to 5,000 tokens. So by the way, what does it mean by the tokens? If you guys have interest in artificial intelligence or the machine learning, then probably you will be familiar with the word token. Furthermore, if you are using the ChatGPT or any other type of large language learning models, then you will be familiar with the word token. But just for the sake of simplicity, please let us tell you that what does it really mean by the tokens here in AlphaFold 3. In AlphaFold 3, if you are going to use a protein sequences, then each amino acid residue present in your protein sequence will be considered as a one token. One very interesting fact that we want to elaborate it here that in AlphaFold 3, you can also model the DNA, RNA, ligands, ion, another type of the post-translation modification in your protein as well. This feature is currently not available in any other type of three-dimensional structural prediction tool. So we hope so that this will make your work very easy. Now, if you want to model the DNA or RNA along with your protein, then the one nucleotide of RNA or the DNA will be considered as a one token. Similarly, if you want to include the ligand in your predicted three-dimensional structure of the protein, then each atom of your ligand will be considered as a one token. Likewise for the ions and the other post-translational modifications. So in total, you can use the 5,000 tokens limits daily. If your model structures are utilizing more than 5,000 tokens, then unfortunately right now, you can't use the AlphaFold 3. So this is one of the most important things that you need to remember. Now let's come back to the AlphaFold 3 server. Okay, we are back to the AlphaFold 3 server. So we got that information that how many tokens we can use daily. After getting this information, now let's come here. Now here you can submit your protein sequences or other type of the sequences which you want to model. So let's copy and paste our protein sequences here for the sake of demonstration. So here we have pasted our protein sequence. Now let's assume for a moment that we want to add a glycan or other type of the post-translation modification in our protein sequence, then what we need to do? In this case, we need to come here and click on these three dots. When you will click here on these three dots, then you will see the PTMs. The PTM stands for what? The PTM stands for post-translational modifications. Please click on that one. When you will click on that one, then you will have this window pop up in front of you. Here on this window, you can select the amino acids or the post-translational modifications. But before moving ahead, please let us tell you that post-translational modifications are not carried out on all type of the amino acid. There are the certain amino acids on which the post-translational modifications are carried out. What does it mean? Let's assume for a moment that you are interested to add a glycan in your protein. In this case, you can only select asparagine, threonine, or serine, because in a natural environment, only these three amino acids in the proteins are carrying the glycans. So let's assume for a moment that right now we are interested to add the glycan here on our serine amino acid. So we will select the serine, and once we will select the serine, we will have this menu in front of us. Here we will select the PTM type. So we will click here on this drop-down menu and we will click on the glycan chain. Once we will do that, then here we have to paste the sequence of our glycan. But question comes here that from where you can get the sequence of your glycan. If you have a good understanding of the chemistry, you can actually draw the sequence of the glycan by yourself. But if you're weak in chemistry, please don't worry. Please click here on this Learn More button and then there will be this drop-down menu. Here you can see the different type of the glycans which are normally present along with our proteins. You can select any type of the glycan sequence from here and copy and paste there. So for example, let's assume for a moment that right now we are interested in this glycan. So we will copy this sequence and simply we will paste it here. After doing that, we will click here on this Save button. But please remember one point here. Once a glycan is added, you can't delete it. So please be careful. Now, after doing that, now let's assume for a moment that you also want to add RNA or the DNA sequence along with your protein. In this case, please click here on Add Entry. Once you will do that, then the new menu will pop up in front of you. In this menu, please click here and then there will be drop-down menu. And in this drop-down menu, select the DNA or RNA in which you will be interested. Let's say that right now we are interested in RNA. So we will click here on this RNA and then here we will type the RNA sequence. Now our RNA sequence is pasted here. Now let's assume for a moment that you also want to add an ion in your modeled protein. In this case, please click here once again on the Add Entry and then click here on the ions. When you will click here, then you will have this menu in front of you. Now click here on this drop-down menu and select the most appropriate ion which you want to add in your model protein. Here, the AlphaFold has provided almost all naturally occurring ions, so you wouldn't find any difficulty here. So let's assume for a moment that we want to introduce the zinc. So we will click here on the zinc and here we will define that how many copies of the zinc we want to have in our protein. Let's assume that we are interested to add the 2 copies of the zinc, so we will click here on the 2. After doing that, let's assume for a moment that you also want to add one ligand in your protein. In this case, please click here on the Add Entry. And after clicking on the Add Entries, there will be one new menu in front of you. And in this new menu, please click on the ligand. Once you will click on the ligand, so here there will be a menu in front of you. Please click here on this drop-down menu and there will be a list of the ligands in front of you. Please select the most appropriate ligand in which you are interested. Let's see that right now we are interested in ANP. So we will select the ANP. After doing this, we will click here on this Continue and the Preview job. Once we will do that, here we are going to have the preview of our job. On the top of this page, you can change the name of your job. So let's say protein modeling. After setting the name, if you are comfortable with all of these details, please confirm and submit job. Once you will do that, your job will be launched and will be in a queue. And you can see your queue job here at the bottom of the page. As the job will be completed, then you can download the protein structure and use it for your downstream bioinformatic analysis. So let's wait here for the completion of the job. Now as you can see it here that our job is completed. Now to visualize the job results, please click here on these 3 dots and then click on the Open Results. Once you will do that, then you will be redirected to the new page, and here on this new page, you can see your predicted 3-dimensional structure along with the other details. As you can see it here that here is your actual protein, and along with the protein, you can see here the glycan attached. Along with the protein and the glycan, you can see these 2 balls. These 2 balls are going to represent the zinc atoms which we have added. Now, along with all of these 3, you can see here a small ligand attached. This is the AMP or the adenosine monophosphate. On top of this, you can see the PLDDT score. You can see the PLDT score in the form of the colors. The blue color represents the high confidence score. What does it mean? It means that if you are going to have a blue colors in your protein, it means that there is going to be a very high confidence on the prediction. The PLDT score between range of 90 to 70 is represented with this aqua color, as you can see here on our protein structure as well. This aqua color is letting you know that the percentage accuracy is between 90 to 70. If you are going to see the yellow colors in your predicted three-dimensional structure, then it means that its accuracy is between 70 to 50. And if you are going to find out the orange color in your predicted three-dimensional structure, it means that its predicted accuracy is less than 50%. That's how you can judge the three-dimensional structure prediction accuracy. If you want to download that three-dimensional structure in your computer for the further downstream bioinformatic analysis, then you can click here on this download. And once you will do that, then the structure will be downloaded in your system. As you can see it here that the protein structure is downloaded in a form of a zip folder. So let's extract it. Now we have extracted the downloaded folder. Let's move into this folder. Now if you see inside this folder, then you're going to find out the different files. Among these files, the most important files are the CIF files, as you can see it here. There will be the 5 CIF files. These files contains your predicted three-dimensional structure. These files will have an index of 0, 1, 2, 3, and 4. The structure with an index of 0 will be the most reliable structure. You can click on this structure, and if you have a PyMol installed in your system, then you can see its three-dimensional structure in the PyMol, as you can see it here right now in front of you. We hope so that now you will be comfortable to use the AlphaFold3 by yourself. In case of any questions, please don't hesitate to contact us. But before leaving, please let us tell you that you can also use the AlphaFold3 to model the multimeric proteins. What are the multimeric proteins? The multimeric proteins are actually those proteins which are going to have 2, 3, 4 or the multiple protein chains. If you want to model the multiple proteins, then you have to select the proteins twice, thrice, or 4 times, and then you have to paste its sequences in AlphaFold3. The AlphaFold3 with a very high reliability predict the multimeric three-dimensional structure of the protein for you. We hope so that all of that information will be very clear to you. Stay with us and we will bring the more informative bioinformatic knowledge for you.